Right, we ready? Cool. Hello. So, thanks a lot for being here and for the organizers to put together this oral session. I'm Alessio Fracasso, chair of this session, together with Professor Lars Muckli. And without further ado, we're going to start with the first presenter, Samuel Bianchi, and the title Assessment of Hemodynamics at 5 frames per second using T-hex spiral in trajectory at 70.
Good morning, everyone. I'm very happy to be here. What I'm going to talk about is it's kind of a nice fit that I can start this session because I'm also going to talk about a lot what comes before modeling activation, which is actually technology that helps you to find better models or model activation more accurately. So what you can see from the title of this talk, we're going to definitely look at the assessment of hemodynamics, namely at measuring the hemodynamic transfer function at different locations in brain. However, it also has this aspect of the T-hex spiral in trajectory, and I'm going to present to you as well what that is. So roughly speaking, what I can already say, it is a trajectory which helps you to scan very fast or highly resolved. Here we decided to go fast. What we did first when we were looking for application of this, of course, we consulted literature a little bit. And there were two papers which were quite inspirational for us. The first one that I want to point out is this review by Jonathan Polimeni. And one figure they have in this paper is where they basically presented oscillating stimulus to participants in the scanner and then looked at the magnitude of the response to that stimulus. And what you can basically see at low frequencies, this magnitude is well predicted by the canonical HRF. However, if you go to higher frequencies, you see that it's massively underpredicted by the canonical model. And what you can directly deduce, if you, for instance, want to resolve this effect at 0.75 hertz, Nyquist sampling theorem basically tells you directly you don't need a TR of 0.67 seconds or below to accurately resolve this effect. So that's sort of hinting us that going fast could be beneficial for modeling the hemodynamic response function. A second paper that we found is one by Logan Dowdle, and he actually measured the hemodynamic response function using an FIR model. And he measured it during two different tasks. One was a fixation task, and the other one was a phase detection task. And what he could actually show at the TR of 1, what you can see on the bottom indicated by those pink or violet arrows, is that at a TR of 1 second, they weren't able to accurately model differences between the HRFs in those two tasks. However, if they lowered the TR, these differences were nicely recovered in the data. So this basically told us, as I already said, modeling the HRF can benefit from a fast sampling rate. The technology we had laying around in our lab or developed in our lab was one done by Maria Engel as a big part of her PhD. And she developed those T-hex spiral in trajectories. And the name from this trajectory comes from, first of all, that is a spiral in trajectory. So you can see I coded the sort of acquisition speed or the time passing during acquisition in the color that goes from blue to yellow. And you can see the trajectory starts in the K-space center, directly moves outwards, and then circles inwards. And while doing so, I think I can show this with this. You see here in these areas, you see those parts where the trajectory is flipping up and down between different planes of the K-space we're covering in this shot. Very briefly, why we do think spirals are a decent pick for fast acquisition is just they have a very high scan efficiency. So you can cover more area or the equal area as an EPI in shorter time. That's why we decided to pick those trajectories. The second thing is if you would place a plane at kx equal to 0 and just look where this trajectory is penetrating through this plane, you will find a picture that looks like this. And that's where the other part of the name of the trajectory is coming from. We call this a tilted hexagonal grid or T-hex grid. The benefit that you get, oh, by the way, the shot that I was showing before is just indicated here in red on the bottom, so we need 5 shots to cover the whole 3D K-space. And this grid has this tilted hexagonal grid, is sort of uniformly dense spaced in K-space. And this dense uniform spacing in K-space leads to a wide spreading of aliases in image domain that is also uniform. It's sort of a caipirinha kind of idea behind this. And that poses the inverse problem we need to solve during image reconstruction quite nicely. So as you can see on the right, sort of the 2 really interesting specifications that we have is that we have quite a large field of view, which we are able to cover, and we cover it at 0.2 seconds per volume. Of course, this needs a little bit of more effort during image reconstruction. So what many people are used to, and what was invented by Klaus Prisman, is the classical sensor reconstruction, where you basically measure a coil sensitivity map before you actually scan. You write down a signal model that includes the coil sensitivity, and sort of in the exponent of the Euler number, you see the trajectory, and you can then just discretize this signal model in space, and you get a linear system of equations which you need to solve to actually get to your object rho here. How we extended this model was by 2 aspects. The first one is the off-resonance map we also measure during a pre-scan, and that basically just tells you, informs the model we are inverting by slight changes of the resonance frequency in the object, because if you place the object or the brain in a 7 Tesla scanner, it will, as a reaction to the strong magnetic field, it will build up its own slight magnetic field, and you need to account for that in the signal model. The second thing, which is shown on the right, is this field camera that we're using. And that field camera is basically just equipped by magnetic field sensors that measure the gradient fields which are actually in the scanner. We do this concurrently, so this can be fitted around the participant's head while he's in the scanner, and we can measure the encoding fields that are around and put this also into our signal model and invert all of this again. Maybe just briefly to give you an intuition that this sort of produces reasonable images, I just showed the mean image of 3,000 images I could acquire in 10 minutes with this technology. So you basically see on the right, we see structures like the cerebellum or ventricles. There's also some great white matter contrast in the images. So we were sort of happy enough with this image quality to proceed to analyzing the data and looking for fitting models of the hemodynamic response function. So maybe very briefly, just where we are now with this technology, I found this paper from 2014 that shows where fMRI used to be back then. And as you can see, I marked where we are in terms of spatiotemporal resolution now. So we're basically at a factor 10 higher than what is indicated here as classical fMRI scanning speed, and still at a quite attractive resolution. The task the participant had to perform in the scanner is a very simple one. We showed happy faces in the center of the screen and happy or sad smileys on the left and right, and sort of the participant then had to push a button with the hand that fit the emotion in the screen. So here he or she had to press a button with the left hand because the smiley on the left is happy and the face in the center is happy. I'm presenting here quite early results, so I only have single subject data at the moment. We're working on getting more data in, but that's what I have. So the analysis of the data was then done as follows. We first started with a GLM approach, where we used the classical informed basis set to use t-tests to find active regions in the brain in relation to our tasks. And based on those activation maps, we identified regions of interests that were 4.2 millimeters spheres. And in those regions, we basically averaged the parameters of the fit to get a measure of the HRF. So you already get a measure of the HRF from the informed basis set. However, then in those regions of interest, we repeated the same thing and just used a very flexible FIR basis set. And because of the high temporal resolution we have, we could actually use one with 160 bins, so 163 parameters we could use to fit the HRF. And we tried to see whether we could spot some benefits of the rapid sampling time that we have. So without further ado, I'm first going to show you some T-score maps, just maybe to convince you a little bit that this produces reasonable activation maps. So this is now the T-score map in relation to button presses, and we found activation in the primary somatosensory and motor cortex, which is what you see on the right. And we also find activation in the cerebellum. So all of this is not a surprise to us. That's basically where we were expecting to find activation in relation to the button presses the participant did during the task. And it's exactly in those 6 regions, so left and right hemisphere as well, of course, where we placed the FIR model in the GLM and looked for and tried to measure the HRF in this way. So here I'm showing the results in the left primary motor cortex. And one thing that we could see here already is that the primary response as measured by the FIR basis set decays faster from the peak than it is measured by the informed basis set. And that's already an effect that sort of requires slightly higher sampling rate because sort of fast changes in signals are usually high frequent. If we then went on to the left primary somatosensory cortex, we didn't find this sort of faster primary response, but what we saw is this very pronounced undershoot period in the HRF, which is again nicely captured by the FIR model because that model just stays very flexible over extended period of time, so we could really easily measure this effect as well. Then in the right hemisphere, I want to be a bit careful with my wording here, so I'm going to say, as you can see, there's sort of this pattern that resembles the initial dip at the start of the HRF. However, at this stage where I only have single subject data and I did a very early analysis, I don't want to say that we found the initial dip. All I want to say is we found a pattern that resembles the initial dip. And what's kind of nice, thanks to the fast resolution, we really get roughly 5 samples that show this pattern, which is nice. We actually find the exact same pattern in the right primary somatosensory cortex. Yeah, that seems to be consistent in the right hemisphere. And we then also looked what we found in the cerebellum. And what you can see here is that the response function just changes quite a bit. It gets much more wider. However, this is something the informed basis set could capture really, really nice. So this informed basis set has basically been designed to really sort of measure a bit widening of the primary response, and this is also reproducible in the right hemisphere here. In summary, I hope I could convince you that 3D T-hex spirals are a powerful approach for doing this type of analysis. It's really nice that we can do whole brain imaging at a high frame rate, which just gives you access to many different brain regions in one experiment. So we could also try to look at interaction effects and those kind of things. And at the moment, I think our results are sort of consistent what it's known from literature, that the HRF varies between brain regions. What I do think is a big benefit is really this high time series fidelity. As we saw at the start, the HRF shows stronger differences from the canonical model at high frequencies, and those are just easy accessible with a high sampling rate, and you can also use extremely flexible models, just thanks to the large number of samples that we have. And yeah, those models, as I said, they just can capture a lot of effects that are around. They're like pretty unbiased towards one specific idea of how the HRF looks. So with that conclusion, I can finally come to my favorite slide, which is the thank you slide. I want to thank my collaborators from the Translational Neuromodeling Unit, Jakob Heinzle, Stefan Fressle, and Klaus Enner-Stefan. And I also want to thank the people from my group, Maria Engel and Klaus Prissmann, who supervised me during this project, and still the ongoing work with it. And last but not least, I would like to thank all of you for being here this morning. It's very nice that you took your time to actually come to listen all of the speakers of this session. Thank you very much.
Thanks a lot, Samuel. We are a little bit pressured with time, so we're gonna move forward to the next talk. So the next talk is presented by Ulrike Horn, Bold Responses in 7T fMRI of the human spinal cord on a single subject level.
Yes, thank you. All right. Yes, you will see that 7T spinal cord fMRI also is for us quite a new technique. So we also have to ask these very basic questions, whether we even see what we expect to see. And so I'm in the pain perception group, so I'm interested in the question, what does pain perception mean and where can we even measure it? And we know that pain actually starts as a signal in the nociceptors and then travels through the spinal cord up to the cortex. And so we know in detail that the first point where this signal enters is the dorsal horn of the spinal cord, and then the signal is relayed via thalamus and brainstem relays to the cortex. Oops, that was a bit too quick. And so the very first point where we would expect some processing and maybe even modulation of that signal would be at the spinal cord. But actually, spinal cord imaging hasn't been there for such a long time, so we still have to, develop a few things here. So because spinal cord imaging actually comes with a few challenges. So as you can see here, the spinal cord has a very small cross-section in comparison to the brain. And within that cross-section, we would like to distinguish different functional regions. So for example, the dorsal horns presenting sensory function and the ventral horns implementing motor function. And of course, we would like to distinguish gray and white matter. So that's quite tricky here. And so we need to be concerned about the partial volume effects here. And so what people usually do in spinal cord fMRI then is to use a very low, very high resolution for in-plane, so that you have about, let's say, 1 × 1 millimeter for fMRI studies. But overall, we deal with a low signal-to-noise ratio. So we would think maybe moving to a higher field strength, like 70, we could actually benefit from that. And so people have done that very recently, and we can now move to more fine-grained anatomical investigations of the spinal cord. People have characterized resting state connectivity already at 7T and did some T1 and T2 star mapping. But of course, every time moving to another technology and moving to higher field strength, this comes with its own problems. And so the first point is, up to now, we have no agreed upon coil design yet. So there are only a few custom-built coils, and so methods are still developed in that direction. The next field inhomogeneities present a significant problem for the spinal cord, because the different tissues with different susceptibilities are very close to each other. And then something I want to highlight here is, so the impact of physiological noise is quite severe and can even be increased at higher field strength. So here you see an extreme example of a very deep inhalation and exhalation. So you see that respiration actually causes some, due to the B0 shifts, it causes some apparent motion in the whole image. And this is obviously pretty severe here. And so how did we deal now with 7T fMRI? So first of all, we now use a custom-built spinal cord coil with 24 channels that also wraps around the neck. To correct for the mentioned artifacts, we need to record respiration and pulse. And to also correct some inhomogeneities in the field, we now use these susceptibility-matched pads around the neck. If you want to know more about these, you can also check out the poster today. And then to induce actual pain in our participant, we use heat pain with an MR-compatible thermode on the arm. And we use a very robust block design to be able to produce robust activations. So we use a 30-second block design. And in that, we have an elevated temperature, which is already quite hot. And then we even have some small peaks to prevent habituation. And so this is individually calibrated temperature, so that participants rate that as painful but not unbearable. And additionally, we now start to also acquire more data in terms of autonomous nervous system responses and psychophysiological responses. So we record additionally skin conductance, and indeed we see some responses to our painful stimuli here, especially at the onset of this 30-second window. And so now to the MRI data. So first we acquire some structural T1 weighted images with the resolution of 0.75 millimeter isotropic. And in that we can nicely segment the cord in there. So that's needed for registration purposes. And we also use higher resolutions even, so about 0.4 millimeter in plane, to be able to nicely segment the gray and white matter here. And so the real challenge are obviously the functional images, because these are way more prone to signal dropouts and distortions. And we try to mitigate that with the parameters listed here. And so we have a very small slice stack here, so it's centered at the level, spinal level C6, where we would expect our activation to be if we stimulate the arm. And in this exemplary slice, you can see our target region, the left dorsal part of the spinal cord. And for preprocessing, we use slice-wise motion correction, which has been developed for the spinal cord. We also include these motion parameters into our models and also automatically censor outlier volumes, for example, due to swallowing. We also use multi-step nonlinear warping to a template space. Statistical analysis is pretty straightforward, so FSL feed. But importantly, we have to deal with all these noise components by using some slice-wise physiological noise modeling, which is also specifically developed for spinal cord fMRI. And so here you see data quality of one participant in the EPI data, but we have to admit here, data quality actually differs a lot across participants. So here you see another participant. And so based on the anatomical images, we know where the spinal cord should be. So I don't know whether you can see it. It's marked here in green. And you see that there are clear signal dropouts and distortions. For example, in this slice, that's the one that's also depicted below. And so in the beginning, when we sent in the abstract, we had gathered data of 4 participants, and 2 of them actually had adequate data quality, and we could analyze the data of the pain experiment. But in 2 of those, data quality was definitely not adequate, and we had to discard that. But the good thing is these distortions are actually mostly due to anatomical reasons. So that means this would be reproducible across sessions. And so that's why we now implemented some sort of screening procedure in which we invite participants once and first determine their data quality. And if they have adequate data quality, then we invite them into our pain experiment. And so now we scanned 51 participants, 70 spinal cord MRI here. And of those, we now selected a few which we could invite. And of these, we now scanned already 9 in our pain experiment. And so that's the data I can present now. So these are, you can see, it's definitely adequate data quality we have now. And so these are the single subject results now. So we will look into the dorsal part, as I said, dorsal part of the spinal cord here. And so in 5 out of 9 participants, we actually see activation there. But you can also see that's quite widespread as well. But this is what we would expect also from 3T spinal cord fMRI. And in 4 out of these, we don't see anything yet. Data quality in terms of like SNR, TSNR is quite comparable. So that's not the point here. We actually noticed that some of these, in some of these, we used lower temperatures. So this could be a reason for this discrepancy, but of course it's preliminary data here. Yeah, and so I hope I could show you that fMRI of the human spinal cord at 70 still faces many technical challenges, but we are now at that point where we can actually see single subject bold responses to painful heat stimulation. But of course, now we have to investigate where do these inconsistencies come from across the different participants, and how can we improve that? And so the next step would be to evaluate different distortion correction algorithms, maybe to allow more fine-grained response delineation. And for us as a group, a very future step would be to have adequate data quality to even investigate the interplay of bottom-up and top-down factors in pain perception in the different layers. But that's way in the future now. So with that, I would like to thank especially the very supportive spinal cord community and also you for listening.
Thanks a lot. So we have time for one question. Somebody from the audience that would like to ask a question?
Yeah, I mean, so we have some ideas about that. So it could be, so there are many different tissues very close to each other. And so it could be, for example, like the bone, how close the bone is, and how close, for example, the spinous processes are, and the different ligaments in between. So the good thing is we now have that data set where we could actually check in like 50 participants which are the factors that determine the data quality. Yeah. Yeah, so you can definitely see that it has some sort of pattern, that it always is at the level of the, where the, how's it called? See, now I'm missing one. We can talk later also about that, but it has definitely some, related to the bone structure, you can definitely see a pattern. So yeah.
Okay, thank you very much, Ulrike. And next speaker is Katarina Marakova, and the title of her talk is Characterizing Coarse Scale Orientation Biases in Human Visual Cortex via Forward Modeling Approach. And Katarina is from the University of Glasgow.
Right, so thank you. It's a great pleasure to actually present this work the first time here in Glasgow, where all of this work actually was done. So this work was heavily inspired by the field of neurophysiology, in which visual receptive fields have been described with many different types of models. So from more simple models, if the receptive field would be described as a simple excitatory function, in later models, it has been shown that with the use of reverse correlation techniques, the properties of visual receptive field firing can be described with two-dimensional Gabor filter functions. And in particular, in Ringach's work in 2002, there has been a division of receptive field shapes described by these Gabor filter functions into even symmetric and odd symmetric, which corresponds to the face of the Gabor filter function. Now, in functional magnetic resonance imaging, there has been some attempts to capture these properties, which can be seen at the neuronal level at the resolution of fMRI, that is at the single voxel level. So one of these models is the population receptive field modeling, which was introduced in 2008, in which the population level response is described as a two-dimensional Gaussian function, which is excitatory, and it's described by its size and position in the visual space. Now, quite recently, in 2012, Zuiderban has also shown that some properties of receptive field firing, such as center surround organization, can also be captured at the level of fMRI, and some qualitative improvements are obtained when fitting this data. Now, when we look at the data that we obtain, when we show participants standard retinotopic stimuli, it can be characterized by many different types of models. So as I said, in the conventional PRF model, we would have a simple excitatory circular Gaussian function, which would give us some prediction of how the signal would look like. Now, what we wanted to know is if we can take the findings from neurophysiology and apply a Gabor filter-like population level response at the level of a single voxel. So in this case, we would have 3 additional parameters in which we could look into the distribution of these at the voxel level, which would be the angle, spatial frequency, and phase. And moreover, we could look how these link to the distributions of the even symmetric and odd symmetric Gabor filters. And in this talk, I'm going to focus on the relationship of angle and phase. So first of all, how did we model the data? So we followed the conventional approach of PRF modeling, but the only difference was that our PRF was now modeled as a two-dimensional Gabor filter function, which had these 3 extra parameters accounting for the angle, spatial frequency, and phase of the PRF. And we calculated the overlap of this PRF with a stimuli sequence of moving bar stimuli, which is the standard retinotopic moving bar stimulus, which for then we would obtain the predicted time series for all of the different combinations of Gabor filter parameters, which we could then fit to the data which we collected with the use of these moving bar stimuli. For the more technical details, feel free to ask about it in the question and answer session. And let's go into the results. So first of all, we wanted to know how do the results compare with the standard conventional PRF model. And as you can see, we can reconstruct the retinotopic maps quite well. We see some deviations later in the visual system hierarchy. Secondly, we can also recover the size by eccentricity relationship, in which the size increases with eccentricity. And there are some deviations in the intercepts and the slopes between the two models, but if anyone wants to follow up on this, again, you can do it in the question session. So then we wanted to look at the quantitative performance of these two models. And so here we fitted both the conventional or simple model and also the Gabor filter model. And for the Gabor, we split the data between the data that was fitted with even symmetric Gabors and the odd symmetric Gabors. And what we actually see is this hierarchy in which the voxels, which were fitted with even symmetric Gabors, had the highest level of performance, then followed by the ones fitted by the conventional model, and lastly by the odd symmetric Gabors. And we can also see some qualitative improvements in the fit of the models. So this in blue, we see the prediction based on the simple model. In red, we see the prediction based on the Gabor filter model. Here are examples from visual area of V1 of one participant, and we can actually it might not be V1, it might be 3. I confused. Sorry, I apologize. But we can see that with the arrows that there are actually some parts of the bold signal which we can capture with this model quite nicely, for example, the negative parts. Right, so after we have established this, we wanted to know the following. So we wanted to look into 3 questions. The first one is if there is any spatial organization of any of the edit parameters. So for example, angle or phase. We wanted to know if these patterns emerged due, for example, a modeling bias, or if it might be driven by a true physiological effect. And lastly, we wanted to know if we can recover the same patterns using a different type of visual stimulation. So let's go with the first question. And so here I'm showing data which have been fitted with or filtered for the voxels that were fitted with the odd symmetric Gabor filter PRFs. And these are voxels plotted in the visual space. And this is data for one participant, only for area V1. Now I'm correct that it is a V1, for sure. And the color corresponds to the orientation parameter of the fitted Gabor filter PRF. And for this one participant, we already start to see some sort of pattern to emerge, which gets more pronounced if we look at the results pulled across all the analyzed visual areas, which would be V1, V2, V3, V3A, V3B, and HV4. And when we look at the data across all participants and all the areas, we start to see this circular pattern in which we see a full representation of orientation from 0 to 90 to 180 and so on. Now, how does this pattern look like for the even symmetric Gabor filter PRFs? So once again, this is data for one participant. Filtered for V3, and the orientation now ranges between 0 and 180 due to the even symmetry of the Gabor filter. And once again, we start to see slightly different pattern. Then this gets more pronounced when we look at the data for all the visual areas. And when we look at the data pulled also across all the participants, we see more of this quadrant bias in which in the The quadrants, which are in green, we have Gabor filters centered at around 45 degrees, and in the purplish blue quadrants, we have Gabor centered at around 135 degrees. So what this seems like is that the orientation effect happens to be phase dependent on the shape of the Gabor filter function. And we can also see these results at a single subject level. So here are examples of 4 subjects. And we can also cross-validate it at a map level. So what we wanted to know next is if this emerges potentially due to a bias in the modeling pipeline or maybe something physiological. And for this reason, we created simulations of PRF responses which had a random distribution of parameters. So this was our null distribution in which no orientation tuning was present at all. And we fit exactly the same model to the simulated data as we fit to the data we collected with fMRI. And we wanted to know if we see any patterns to emerge. And in the case that we would see the same patterns, it would highlight that there is a modeling bias. If not, it might be something physiological. So let's just jump in. And so we can see for the effect that we see with the odd-symmetric Gabor filter PRFs, it actually emerges in the simulations. And we see that all the different types of noise that we use, which highlights that this effect really does seems to be driven by something which is present in the modeling pipeline. And if anyone wants to follow up on this, please feel free to do so again in the questions. And for the ones that are fitted with the even symmetric Gabors, it does not seem to case. So this doesn't show up in the simulation, and it might be something physiological. So what we did finally, we wanted to know if we can recover this using different type of visual stimulation. And for this reason, we downloaded the data from the Human Connectome project. We selected only the runs which have been collected using the rotating wedges and expanding and contracting rings. And we fitted exactly the same model. And what we actually see is both of the patterns. So this type of stimuli is still sensitive to the modeling bias as shown before. And also we obtain this quadrant bias, which might be actually driven to something by something physiological. So just to summarize, we see with this new model that there is some coarse-scale topography of orientation tuning using this model. We can also see that one part of it seems to be driven by a modeling bias, whereas the other might be driven by something physiological. And we can also recover this using different type of visual stimulation, which means that perhaps the effect that we see for the even symmetric Gabor might have some link to the literature on radial bias, such as the one introduced by Sasaki in 2006, or it might have something to be with optic flow, but we're still currently speculating on what this might be. And I would like to thank my supervisor, who happens to be chairing this session, and the postdoc who was heavily involved in this project, Dr. Jasper Fabius. And thank you for your attention.
So Alessio is the supervisor mostly, so therefore I can ask the question. If there's any question from the floor, otherwise we have one online that is, do location XY parameters for each voxel typically match for simple and Gabor models, or do you find a systematic shift?
So they seem to be quite similar. We haven't really explored it that much, but I think the best way to look at it is based on the retinotopic maps. Since there is the confound of this model that we see here, because it's confounded, the locations tend to be fitted with more eccentric and larger PRFs, which also then makes as I showed before, that there is the deviation later on in the visual hierarchy, which is mostly driven by this. So otherwise, the positions are very similar or comparable, I would say.
Thanks a lot, Katharina. We're moving on to the next talk. The next talk is from Raphael Modek, from Paris, and it's called Comprehensive Brain Reading Decoding. Mental processes from web repositories of brain images. And while we're waiting for the slides to come online, we had one more question for the previous talk. No, for the one before, actually. For Ulrike. So, question for Ulrike. Is the subject's selection biasing the analysis by who has a good enough anatomy to get consistent group analysis?
Oh yes, some people were asking me this as well, because my sample actually consisted, for example, of more male. And so we thought, I mean, with 9 people, we couldn't really tell whether there are systematic differences. For example, with the male-female ratio, we noticed that, for example, when males are on average larger, then the lungs were not as close to the area which we measure. And of course, the distortions highly depend on that. So most of the respiration-induced artifacts would then be even stronger in smaller people. But yeah, other than that, I think it's beneficious if, for example, the cord is very straight, because then, I mean, we don't have that much of resolution in that direction. So that would also be a factor. But other than that, we try to investigate exactly that, which are the factors that determine data quality here.
Okay, thanks. And then there's another question. Can we replace activation models like GLM with machine learning approach?
In my kind of data? So.
Yes, well,
yeah, I mean,
I think it came into your time. It's hard to tell, but
okay.
It was okay. Yeah. Okay.
All right, we're going to try to plug the computer directly, see if I'm luckier with the presentation.
Sorry about that.
Cool.
Okay, so I think we're all set. So thanks for the opportunity to present my work. So we are going to talk about decoding cognitive processes from large-scale data using multiple studies. So quick refresher on fMRI data and task fMRI data. So we record the bold signal of a patient while he is under a stimuli or doing a certain task. And from there, we can create statistical maps where the regions that are specific to this task are visible. And what we have an interest in is the decoding part here, which is we want to, from a statistical map, we want to infer which cognitive processes have been implied in the experiment on the subject. So before diving into the details, there are 2 projects of open data that has been crucial to our work. So the first one is NeuroVault, which is an open database of fMRI data. And the idea is that users can upload fMRI maps along with some labels or description. And to this day, it contains around 200,000 images. And this allowed us to have access to a very large number of fMRI maps. The other project that has been useful is Cognitive Atlas, which is a collaborative project to create an open database of cognitive neuroscience. So it contains cognitive processes along with the tasks that allow to measure them.
And what is of interest for us is that everything is organized in a hierarchy, and it will be useful in this situation. So what we do is decoding, and we do that in the multi-label setting. So for a given map, we want to predict potentially multiple elements that have been involved in the experiment, and not only a simple classification. So we did that by in two parts. First, we have a processing of the images and the fMRI maps, and we did also some curation and pre-processing on the label side. And I'll go through both sides. So if we start with the image space, we basically did some selection of fMRI maps from NeuroVolt. So basically, we selected the Z map and T maps, and also put aside some maps that presented some weird values, non-values, 0 values everywhere. And from the around 50,000 images that we selected, we applied some common resampling, and we put them in on the Diffumo atlas. So Diffumo is a probabilistic atlas with potentially a large number of components. There is multiple scales to it, from 64 components to 1,000. So it allowed us to drastically reduce the size of the data in memory to a simple matrix of 50,000 maps to 1,000 components. On top of that, we used three neural networks with 3 hidden layers, which is pretty simple. We applied some penalization and dropouts for the learning process, and we output multi-label prediction, as said earlier. So this gives us nice results. We initially decode around 35 concepts with large gains compared to previous methods. And the gain is also that it's a wider spectrum of decoded elements. So here what you see is the AUC, so chance level is around 0.5, is not around, is. And we see that for most of the items, we decode with a great score compared to the chance level. So I talked earlier about doing some curations in the labels. So one strength of NeuroVolt is that you have access to a large number of maps and the labels that are being uploaded to it by the users. But the problem is it lacks some general roots, and there is some differences in how people annotate the images. So basically, we used Cognitive Atlas to curate a bit the labels that are in NeuroVolt. So basically, we know from this hierarchy that's a map, for example, which has been tagged by the user as speech perception. We can apply already the voice perception tag and follow the hierarchy of tags that are in the cognitive atlas. So it allowed us to enrich a bit and have cleaner common tags between images. So this leads to a decoding of around 50 decoding cognitive processes. Note that we process in a collection leave collection out a pattern. So basically here what you see is the results on IBC, where IBC has not been in the training set. So we train on different studies that are in NeuroVolt and evaluate on newly seen IBC, and we obtain again successfully some decoding of the 50 cognitive processes. One thing we note is that some elements that are largely in the training sets, such as the working memory, are poorly decoded, and this can be a sign of the different definition in the in the community of what the working memory. Okay. What the working memory would be. So basically, it shows different realities. So we looked after at the sensitivity maps. So basically, we derived the outputs over the inputs to generate those maps. And what we see is that for well-decoded terms, we obtain maps that are really expected when compared to what is in the literature. When maps which are poorly decoded, such as working memory, doesn't represent any pattern that we expect, and it goes. We looked at also some elements. For example, in the left-to and right-to decoding or syntax, we see regions that are kind of expected, and we also noted some biases in the data. For example, in the emotion perception, where it relates more to faces than to emotion perception. And this is probably due to the task being recognizing emotions on faces of people instead of mainly, which leads to this bias in the maps. So if we conclude, so we leverage open initiatives to apply large-scale decoding in a cross-studies way. And we successfully decoded around 50 cognitive processes. And the idea is we want to share is that there is value in sharing data online on Neurovault because there is a use to having those large databases of fMRI maps. And one downside we saw is that the labeling quality is still something that the community needs to work on, and there is still some efforts to do on there. So if you want to discuss more, we have a poster at the poster station just right after.
Thanks a lot.
Okay, do we have some questions? Yep.
We have a question over there.
Your push for getting people to share more data on Neurovault, is there any sort of preference for having thresholded versus unthresholded maps? I feel like this is something that we've debated in our lab.
I guess I'm kind of fairly new to the community. I'm a PhD student, so my guess would be to share the unthresholded map because it gives you share your data, but you also share your experiments, and you can share your analysis on one side on how you thresholded the maps and what is the pipeline you did to the experiment. But I think it's nice also for everyone to have access to the raw maps and potentially explore by themselves without having this limitation.
Thanks a lot. So I think after the technical issues, we can move to the next talk. All right. Let's see if we are luckier with this one. So the next talk is going to be presented by Lauren Patrick. The title is Local and Distributed Colloquial Markers of Effort Expenditure. Thank you, Lauren.
Okay, hello. Today I'll be presenting some of my work from when I was a PhD student at Yale on local and distributed cortical markers of effort expenditure. So while we've learned foundational insights from binary force choice decision-making paradigms, those don't allow us to capture dynamic reevaluation that's necessary in sustained goal pursuit. So if you imagine I give you the choice between choosing the path on the right and the path on the left here, you may initially choose the path on the right, but then you start going down the trail, you realize that that might actually add additional distance to your hike, and at some point you reevaluate and switch to the other path. So this is something that isn't captured in those traditional paradigms and something that's really interesting for looking at sustained goal pursuit. So understanding this continuous reevaluation requires us to develop tasks that more closely mirror these naturalistic types of settings, but it's also important that we're able to look at component processes. So you can imagine not only the physical effort expenditure is important in this case, but also the total time and also potential rewards. So maybe you see a great mountain vista if you choose the path on the right. And as I've hopefully just convinced you, this is a very complex process that requires a lot of different components of cognitive processing. And because of that, in my work, I'm interested in looking at these processes using large-scale brain networks to see how cognitively these processes track with networks. And in particular, in the work that I'm presenting today, I'm interested in the role of default network, which has been implicated in many processes, including containing canonical reward circuitry, and also the frontoparietal network, which has been implicated in goal selection, response selection, and performance monitoring. So that brings me to the core question of this study, which was which brain systems support willingness to exert effort during the pursuit of desired goals? And to examine this, I developed a novel task called the Willingness to Exert Physical Effort, or WePhys task. There are two components to this task. The first is the practice phase. Within the practice phase, participants are told to practice pressing a button with their right index finger as many times as possible within a 15-second window to get used to the task. We use the latter 3 of 4 practice trials as a titrated value to just get an indication of their baseline ability to button press. And you can see on the upper right, that's our distribution of titrated values for the participants in this study. And individuals do vary at baseline in their ability to expend effort. And then on the bottom right, you can see the distribution of required effort expenditure. The titrated value is at the center of that distribution, and we build required effort expenditure for each trial around that in a fairly normal way. So then in the main component of the task, individuals have 15 seconds to press the button as many times as they would like in order to increase the likelihood of obtaining reward or avoiding punishment. So in this case, they don't know how much effort they would need to expend on any given trial in order to be successful on that trial. There are reward and punishment blocks. And at the end of each trial, individuals learn whether they received a reward or avoided punishment on that trial. Okay, so moving into our imaging results. First, we were interested in looking at whole brain, what tracks with these processes. And in particular, we were interested in looking at total earnings and time on task, and also the condition, so if they're in the reward or punishment condition. So we ran a whole brain ANCOVA with those 3 components, and we were particularly interested in seeing whether medial prefrontal cortex tracked with these processes because it's been implicated in willingness to wait for rewards in the future, but it's less clear what the role is in sustained effortful goal pursuit. So from our ANCOVA, we found that MPFC was one of the territories that tracked with scaling effort expenditure. And from there, we wanted to further look into the time courses and see what that actually looked like over our 15 seconds of the trial for each participant. And we saw that there is a difference between the reward and punishment conditions. So on average, in the reward condition, there's greater activation in that MPFC ROI. In addition, high earners show greater activation in that MPFC ROI, but we didn't see a difference in time in this case. So from there, we were really interested in seeing whether we could decode how much effort someone had previously expended from the feedback portion of the task. So this is when there isn't any active effort expenditure, but we're looking at whether we could decode how much effort they'd previously put in on that trial from that reward phase. And to do this, we use support vector regression. So we use a cube instead of looking at a mass univariate type of approach. And the cube moves through the whole brain, and we build a model based on 90% of the data and then test it on 10% of the data. And we're looking at the relationship between actual effort expenditure and predicted effort expenditure, and that held out 10%, and we repeat that 10 times. So there we found that we could decode previous effort expenditure at reward feedback. It was widely distributed throughout the cortex, which we expected. And that includes aspects of prefrontal cortex, the parietal lobe, both medial and lateral PFC. So from there, we were really interested in seeing, okay, we are able to decode how much effort someone previously put in, but how does that track with these networks that I brought up previously that have been implicated in these cognitive processes such as reward or sustained attention? So we actually found that frontoparietal network had the greatest decoding accuracy. This network has been implicated in such functions as cognitive control. And following that, dorsal and ventral attention networks had the greatest decoding accuracy, with default being right around average despite that, including the canonical reward circuitry, which we would expect to be highly implicated in a process like this. So this is interesting because it extends beyond the ROI approach that we did previously and treats the brain more as a system that functions as a whole instead of only in discrete regions. And just to check to see how our decoding accuracy compared to a null model, we built null models for each of the networks by shuffling the labels, and we found that our decoding accuracy for each of the 7 networks was actually higher than the null model. So we were able to successfully decode from each of these networks, which suggests that this process really is widely distributed throughout all of the networks that we examined. So the conclusions from this project are ventral MPFC activity tracks trial level effortful persistence as a function of time, monetary incentives, and eventual task earnings. Subsequent feedback responses in frontoparietal and dorsal attention networks preferentially reflect prior trial level effort expenditure. And also signals related to prior effort expenditure can be decoded from large-scale distributed brain networks. So with that, I'd like to thank my lab and also my funding from the Yale FAS Discovery Fund.
Thanks a lot. So we have time for a couple of questions. Somebody from the audience? Lars, spot somebody? No.
No.
It was a false alarm. But I have a question. So, are there neurophysiological findings from monkey neurophysiology, I'm thinking, or also mice neurophysiology that are comparable with what you observe in humans? And if so, what are these findings?
Yeah, so that's an interesting question. The homology is a little bit tricky with PFC, but in non-human primates, a lot of the focus has been on DACC. So that's within our frontoparietal network that we were looking at. So that network that's in the orangey color. There is evidence there within non-human primates, and also the striatum is heavily implicated in these processes, reward, effort expenditure, et cetera, and the dopaminergic system broadly. So in rodents, a lot of the findings have been within ventral striatum in particular, and the role that that plays in motivation and willingness to exert effort for reward. But being able to look at the PFC in this way is a little bit different in humans. So.
In what way is it different in you?
Just the homology of territory. So like VMPFC, DACC, it's a little bit difficult to find the exact territory, especially in rodent, for instance, to see how that maps. So
it's very clear.
It is convergent, though.
Anybody else? I don't see. All right. So thanks a lot. So now we can move to the last talk of the session. Is Lina Teichmann, the temporal dynamics underlying behaviorally relevant object properties during visual processing.
All right, thank you. Thank you for the introduction, and thank you everyone for being here. So in the study that I am presenting today, we asked how we make sense of what we see. So if I was to show you these 4 images, you would be very quick and very accurate at telling me that you see a spider, some threads, an envelope, and a rocket. However, these are just 4 categories of many, many categories that you can identify accurately really quickly. So in our study, we sampled from 1,854 different object categories that were taken from the Things database. And for each of these categories, we had 12 different exemplars. We then took these images and showed them to people while they were sitting in the MEG. And in total, we scanned 4 participants who came back for 12 sessions and saw all of the 22,248 images. None of the images ever repeated, so every person just saw every image once. We then recorded their brain activity across the 272 sensors, and they were time-locked to each of these images that were shown on the screen. So the most obvious and first question we can ask here is, when are there different category-level representations evoked in the brain? So to do this, we ran a time-resolved classification analysis where we extracted the sensor pattern at every time point from all of the 270 MEG channels. And then we trained a linear classifier to distinguish responses that were associated with 2 of the categories at a time. So running this across all of the 1,854 pairwise comparisons, we can then look at the decoding accuracy over time. And what we can see here is that there is information about the object category in the neurosignal at around 100 milliseconds. If we look at higher level, like higher order categories, we can look, for example, at MDS and also see compare animals and foods, for example, or vehicles and tools. And then we see that the time point of separation differs for higher level categories. So this is all nice, but really it's unclear from this what visual or semantic properties drive the category decoding. And in particular with this, I mean that you can actually do a lot more with these images than just categorize them. So if I was to ask you to group 2 of the images that kind of are similar, you might be picking the spider and the rocket because both of these can move. Or another person might pick envelope and rocket because they are both white. So getting at these multi-dimensional representations, my collaborators have run a large-scale behavioral studies a few years back where they showed participants 3 images at a time. From these images, they then asked participants to pick the odd one out. So in this case here, people might pick the rocket because the other 2 images shows things that are very colorful. However, if I replace this image in the middle, participants might pick the watercolor because the other 2 images show something that can fly. Using a computational model on these behavioral responses, we can narrow this down to 49 distinct dimensions that kind of drive the behavioral responses of participants. The weight on each of these dimensions indicates how strongly an image is associated with a given dimension. So here I'm just showing you an example dimension 11, and on the left side are some 4 example images that have a high weight on this dimension. Whereas on the right side, we have 2 of the low-scoring images on this dimension. This is just 6 example images, but actually all of the 22,000 images have a score on this dimension. And here's another dimension 29, which shows, again, the high weight on the left and the low weight images on the right. If we then ask an independent group of participants to label these sorted images, we find that for this top dimension here, most people say that this dimension has something to do with colors and whether images are colorful. Whereas the bottom people associate this with something that is flying related. So, but we can also look at this the other way around. So if I go back to the categories, we can say across all of these 49 dimensions, what kind of content is associated with thread. So here, each of the length of the stems shows us how much each dimension is associated with a category thread. And you can see that the top 3 dimensions here are colorful, cylindrical, and handicraft related. For spiders, on the other hand, we have animal-related insects and repetitiveness as the top associated dimensions. So taking this back to our MEG data, we can ask whether these dimensions are accessed at different stages of visual processing. So again, I'm just extracting the sensor activation pattern at every time point from the MEG data. And now just focusing, this is every dot in here just shows one specific trial. And here just focusing on one single dimension, such as colorful, we can then associate each of these trials with how strongly behaviorally we are weighting these on this dimension. I'm then running a time-resolved linear regression for this dimension to see at what time point the dimension is represented in the neurosignal. So to do this, I fit a plane that is describing the linear relationship between the neuroactivation pattern and the dimension weight at every time point. Then I take independent data from a different session, and just extracting the MEG data, we can then predict what the dimension weight is for the data that we extract from the MEG signal. Then to assess how well the prediction matched the true dimension weight, we can then just run the correlation and say, to kind of assess like how well we can predict the dimension at a given time point. If the correlation is high, it means that the dimension is represented in the signal at this time point. So here are the results. So I am, the length of the stems reflects the correlation. And you can see now at 100 milliseconds, some of the dimensions start to be represented in the neurosignal. And you see like some of them are growing and decreasing again. So really the different dimensions are represented at different stages of visual processing. In addition to looking at all of these 49 dimensions at the same time, we can also focus on each of these and just look at the time series in a more kind of classical way. And I just picked 3 here to demonstrate, but obviously we have 49. And you can already see that there's some key characteristics. So for example, colorful has an early peak, whereas food-related and sports-related do not. So to assess how, like, which kind of dimension time courses are similar, we wanted to compare them all. And here, I'm just showing you 2 of them. So this is colorful and cylindrical, and already just looking at these, you can see that they are quite similar. We ran dynamic time warping to see how much we have to warp or change one of the time series to make it look like the other one. And we can also demonstrate this in this cost matrix here. So if so the gray thing is like showing the path of the lowest cost. So if 2 time series were completely identical, it would just fall on the diagonal. So here are 2 other time series, colorful and food-related, and you can already see they are quite different. So if we use dynamic time warping, we have to like warp one of the time series more to make it look like the other one. And the path of the lowest cost, shown in gray, shows here that the distance there is longer. So the advantage of this technique is that we can take the entire time series and kind of narrow it down to one single data point. So we can say how similar are 2 things from each other. So we did this across all of the 49 dimensions. What we can do then is run hierarchical clustering on this and to see which kind of time series have like a similar shape. So here on the left, you can see them in the ordered way. And from our hierarchical clustering results, we have 4 different clusters. And if I just average the time series from each cluster, we have something that looks like this. So we can already see here that there's like some key characteristics. So in some of the clusters, we have an early peak that's either there or it's not. And then it also seems to matter what the relative size is of the second peak. So it is possible that these characteristics are related to whether the dimension reflects primarily a perceptual or a conceptual property. So to conclude, we showed that behaviorally relevant dimensional content can be read out from time-resolved neuroimaging data. We believe that our dimensional approach gets us a little bit closer to understanding the neural underpinnings of real-life visual object processing and the complexities thereof. And lastly, we also showed that we can use our large-scale MEG data to extract prototypical time series patterns. And with that, I'd like to thank my collaborator, Martin Hebert, and our PI, Chris Baker. And if you missed the poster sessions in the last 2 days, check out Philip's poster online where you can learn more about things. Thank you.
Thanks a lot. So we have a couple of questions from the app. So the first one is, to what extent do you think these different time series shapes could be due to low-level visual properties?
Yeah, so this is a good question. This is something that we are kind of starting to look at the moment. So because the images are all like natural photographs, they are not controlled for any low-level differences. So at the moment, we are kind of trying to see which key characteristics, which image properties are driving these characteristics in the time series. I don't know yet, though.
And another question, are the dimensions affected by cultural backgrounds of the subjects?
I mean, that's possible. We only have data of 4 subjects at the moment, but I think there is like another group, I think in Montreal, is collecting more data on with the same images also on MEG. So there may be more opportunities to investigate this in the future.
Thanks a lot. So thanks. Oh no. Oh, great.
Just a very quick question. Do you plan on sharing the data, preferably with MEG pre-processing for people who are more familiar with decoding but not with MEG?
Yeah, so we are going to have a data release paper most likely this year with the MEG data, fMRI data, and more behavioral data that will all come out this year.
Thank you for a great talk. From what I understood, in figuring out which dimensions occur at what time point, you're using all the channels. So, yeah. but different categories we know that are represented in different areas or regions. So different channels have different weights on how. On determining this. So aren't using all channels adds more noise than signal from the channels that are irrelevant to the dimension?
Yeah, that's a good question. So using all of the channels means that, yeah, we might be increasing some noise, but I am assuming that channels that don't carry any useful information in the training set will not really be considered at the testing set. So yeah, I mean, we can go all the way and do source level and like kind of distinguish it at this level, but also we have a full fMRI data set on this, so we kind of plan to ask the more time questions with the MEG and focus on the spatial resolution with the fMRI data set. Yeah.
Thank you. Great.
Why 49?
That just came out of the behavioral model that yeah, was published a few years ago by Martin.
Lars, a question over
there. Thank you for the very interesting presentation. You were able to have a human interpretation for each and every one of the 49 categories, because this is not something that you necessarily enforced in the determination of those categories, is it? So.
I'm not sure I understand. Say again.
So you obtain 49 like those dimensions that can characterize the images, and then you try to annotate humanly all those categories, right?
Yeah, so the labels is also based on what like an independent group of participants have named the images.
And yeah. they were able to clearly identify a humanly understandable label for each and every one of those categories?
No, so like I showed you the word clouds, right? So there's like some variants of what people said. So right now when I label things as the dimension, it's just the most common label that we got. But it's not like there are some things that are like more varied.
Okay, because this is not something that you search for when you identify those categories, right? You didn't look for embeddings that would be meaningful for humans. Interpretation, right?
At the dimensional level?
Yeah, while finding those 49 different
categories. That has nothing to do with the label. So like this comes purely from the odd one out task, where they have the 3 different images and they pick the one that's the odd one out. And then the labeling happened at the next stage. Yeah.
Thank you.
All right, so I think we can call it a day. Thanks a lot for your attention. Thanks a lot for the speakers. It was nice. Thank you.
Kept the time pretty good.
It was good, no?
Yeah. Well done.
Very good. Tight.